We're getting into Unit 3 now, so let's start with talking about the types of cells and how we tell the difference between cells and how we organize them, and then a little bit about their organelles as well. So starting off, let's do a quick little review of the characteristics of life and viruses, because that kind of often gets lumped in with cells, and we're going to be talking about our cells versus bacteria, so we might as well get a little bit into viruses here as well. So be able to determine whether something is living or non-living, describe the structure of viruses, and explain why they are non-living. So we talked about the characteristics of life. Basically, life is made of cells, they reproduce, they have to get energy and drive their own metabolism. They have to be able to sense the environment and respond to it, maintain a constant internal environment, evolve, all that fun stuff. Viruses are just small infectious particles. They can only replicate when they are inside a host because they are made up of DNA surrounded by proteins, and that's it. They have no organelles, they are not cells, and they don't have any type of machinery to obtain and convert energy into a usable form. They have no machinery to copy their own DNA. So they have to hijack living cells and use all of the equipment and machinery in order to get them to live. So one of the ways that viruses are very similar to life is that they evolve. So viruses are often grouped into species in the sense that we group them together based on similar DNA, similar characteristics and structure, and things like that. Viruses are very particular in what species they can infect, and it often comes down to the envelope or what type of coating that they have. And we tend to group them based on what kind of proteins they have in their capsid and that protective protein layer, things like that. So that's just a brief reminder of why cells are not considered living, and they have to hijack our own cells in order to function at all. Now let's get into cells themselves, the actual basic unit of all life. And we'll get into a little bit of cell theory and the different domains of life and all of that. So be able to describe cell theory and why it's important, describe the 3 domains of life and how they evolved. We'll get into how they evolved in class, and then give examples of organisms in each domain, and then describe the different structures of our prokaryotes, eukaryotes, and compare that to viruses. So the cell is the basic unit of life in the same way that the atom is the basic unit of matter. And cells were first discovered by Robert Hooke back in 1665. He took a microscope to some sliced up cork, and he ended up seeing exactly this right here. This is his drawing of the cork cells. And because they looked like little rooms in a monk's dormitory, that's where he gave them the name cell. We could have very easily been calling these animalcules because in 1674, roughly around the same time and independently, Anton van Leeuwenhoek, who was just a merchant but enjoyed making his own lenses and looking at everything he could under that microscope that he created, saw the first living cells, and those are some of his drawings up on the top. So Robert Hooke only looked at dead cells, particularly the cork, whereas Anton van Leeuwenhoek was the first to see living cells that were actually moving around under his microscope. But it wasn't until many years later, in the 1800s, when cell theory was first developed. So scientists had started this microscope revolution. They were looking at absolutely everything under the microscope, and they started to realize that all living things had these cells as part of their structure. So Matthias Schleiden in 1838 and Theodor Schwann in 1839 made the blanket statements that all plants and all animals are made up of cells. And this becomes the first part of our cell theory, that all living things are made of cells. This is where we get that the cell is the basic unit of life. If you are not a cell, you are not alive. Now, Rudolf Virchow was the first to see cells divide in 1855. We'll talk a little bit about this a little bit more in another video about the germ theory of disease, about where we thought cells come from, but basically we just thought they kind of popped into existence from nothing. So Rudolf Virchow, looking under the microscope, actually saw one cell split and become two in 1855. And this is where we get the second part of cell theory, that every cell has to come from a pre-existing cell. No life can come from nothing. Life has to come from pre-existing or already living things. Now, cells have 4 features in common. No matter what type of cell they are, they have to have these 4 characteristics. Honestly, if we went and found life somewhere else in the universe, we would probably have to look at how do they meet these 4 characteristics as well. These are such basic things to what it means to be alive. First, you have to have some kind of barrier that separates self from non-self. Like for us in our bodies, it's our skin. For the cell, it's the plasma membrane. We have to have some type of barrier that controls what comes into the cell, what comes out, so we don't end up just kind of mixing with our environment. Now, because almost all chemical reactions that are required for life require water for them to occur, we have to have some type of water-based liquid that fills up the interior of the cell so that all the chemical reactions needed for that life to occur can occur inside this water-based medium. And we call that the cytosol or the cytoplasm. Those words do mean something different technically, but we often use them very interchangeably in biology. And this is just the liquid that kind of fills up the cell. Now, the next thing that we need is a code to say what type of cell you are, and a code that will drive what proteins you make and how your cell reacts to the environment and how it builds itself. And that code is DNA, and we call that our genetic material. So we need DNA or RNA or some type of nucleic acid that has the code that tells us what to be and how to be it. The next thing that we need is a way to convert that code into working proteins, because the code is great, but the code is just a code. It's like a blueprint. You can't exactly, live in a blueprint. You can't, drive around a blueprint. You have to take that rendering and convert it into something tangible that becomes the structure and the function. So in cells, we have ribosomes to do that. These are special little structures that convert the DNA code into proteins, and we will talk about them much more in detail when we get to our DNA section. So life, as we've been discussing, is highly organized. We've mentioned the organism level and up, from organism to population to community, ecosystem, biome, etc. Well, what about the other way, going down? The organisms are made up of organ systems, such as our skeletal system, our muscular system, our nervous system, and those are made up of organs like the brain, the heart, the muscles, the bones. Those are made up of tissues, which are groups of similar types of cells. Those are made up of specific cells themselves. Cells are made up of organelles. Organelles are just large molecules, often those proteins, carbs, nucleic acids, and fats and lipids that we discussed before. And then molecules are made up of atoms. And we don't really go any lower than that because the atom is the fundamental unit of matter. So let's look into some of this in a little bit more detail now. We also have to categorize life by where it fits in taxonomy. Taxonomy is just a way that humans have looked at all the life around us and categorized it. We call you a plant. We call this, a mammal or a bird or something like that. So the largest level, the highest level, the least specific is the domain. And there are 3 different domains, and they are all based on your cell type. So 3 domains based on cell type. And then we get more specific from there. Then we have our kingdom, our phylum, class, order, family, and the most specific are the genus and species. We put those 2 together to get our scientific names. So the 3 domains of life are bacteria, archaea, and eukarya. Bacteria were the first to evolve. These are the first types of cells to evolve on Earth. They are single-celled organisms, and they probably evolved around 3 billion years ago. And they're made up of a type of cell called a prokaryote. We'll get into that in just a few slides. The archaea are also prokaryotes, and they are also single-celled organisms, but they evolved slightly after the bacteria, and they evolved along a different path from bacteria. They're actually a little bit more similar to the other domain, eukarya, than they are to the bacteria themselves. And eukarya, these are our eukaryotic organisms. I'll say what that means in a moment. And they can be made of one cell or many cells. And this is the most recently evolved cell type. So they're the youngest type of cell on the planet. This includes pretty much all the life that you're most familiar with. Plants, fungi, animals, that kind of stuff. So these 3 domains of life each have 2 different types of cells. There's 2 different types, or the prokaryotes fit into 2 domains, whereas eukaryotes are their own domain. The prokaryotes are often smaller cells. They are more structurally simplistic. That does not mean they are simple at all. They are just as complex and fascinating as eukaryotes, but they have a little bit more simplistic of a structure. They do not have a nucleus. They lack a nucleus. And they have no membrane-bound organelles. They lack those as well. So what that means is all the organelles that we're going to talk about later, they're all made up of the same membrane structure as the plasma membrane, and our prokaryotes, our bacteria, and our archaea do not have that. The eukaryotes are much larger, more complex. They have a nucleus, and they have membrane-bound organelles. So that is the key thing that differentiates the 2 different types of cells. Eukaryotes have a nucleus and they have membrane-bound organelles. Prokaryotes do not have a nucleus and do not have membrane-bound organelles. You want to be tempted to go with their shape and size first, but that's not their defining characteristic. There are some very small eukaryotes and some very, very large prokaryotes that kind of break the rules, but this rule of the nucleus and the membrane-bound organelles is not broken. So prokaryotes have basic cell structures. They don't have a nucleus, but their DNA is found in a nucleoid region. Basically, that DNA just kind of huddles up in a specific area of the cell. And they have 1 chromosome. Humans have 23 pairs, whereas prokaryotes, they only usually have 1, and it's usually a circular chromosome, whereas ours tend to be more linear, and you'll often see them depicted as an X. Bacteria, it's just a little circle. Sorry, I'm trying to erase this, and my pen is giving me issues. All right. Now, they also can often have plasmids. These are much smaller little pieces of DNA that are also circular, and they don't necessarily contain the same information as the chromosome. They often contain extra genes or extra information that help the bacteria survive in different types of environments. They also have a plasma membrane. They have to, they're a cell. And they have the cytoplasm. Once again, they have to, they're a cell. And they also have ribosomes. Those are those little dots right there. Remember that those are what help the cell convert the DNA into proteins. So these are the 4 basic things that every single cell has to have: DNA, plasma membrane, cytoplasm, and ribosomes. But prokaryotes have some extra things as well. They're not just the simple 4 things that all cells have to have. They often have specialized cell walls, and this is different based on the species. There's really, 3 main groups that we tend to look at. One's called gram-positive, one's called gram-negative, and others that are acid-fast staining, often like our mycoplasma bacteria, and those just have like a lot of waxy lipids in their membrane. Now, some species of prokaryotes have capsules. These are really thick layers of carbohydrates that surround the cell and make them sticky. This allows them to stick to parts inside your body or to surfaces and keeps them from drying out. It makes antibiotics harder to attack them, things like that. They sometimes have these things called pili. These are little things that look like hairs on the outside, little proteins. And this allows them to attach to each other or to other structures. When they attach to each other, they can even transfer some DNA back and forth. And then they can also have flagella, this long tail structure, and that allows them to swim and move around in their environment. Now, the eukaryotes, remember, they're larger, they're more complex because they have a nucleus and membrane-bound organelles. However, they still have to have the 4 main requirements of every single cell. They have to have some kind of genetic material, their DNA. They have to have a cytoplasm. They have to have a cell membrane, and they have to have ribosomes. Now, their DNA, what's special about the eukaryotes is they put their DNA in a nucleus. So all the DNA is found inside that membrane nucleus. They also have lots of other tiny organelles that are all made of membrane. And this is what we call compartmentalization. Basically, the eukaryotic cell is different from the prokaryote in that it makes smaller rooms inside itself so that specific reactions can occur inside those rooms and not affect other parts of the cell. You can kind of compare the prokaryotes and the eukaryotic cells to a studio versus a 1 to 2 bedroom apartment. So the studio is where your bedroom, your sink, your kitchen, your bathroom, they're all the same place. Your living room, it's all one room, and you just got to figure out a way to live in that one room. So you're going to sleep right next to where you cook your food. Whereas with eukaryotes, they've evolved to have different rooms. They're like a 1 to 2 bedroom apartment. Your bedroom is in its own separate compartment. Your kitchen is in its own separate compartment. Your living room is its own separate area. Your bathroom is separated. Each of these now has their own function, their own role, separate from all the other rooms. Now, some types of eukaryotes have cell walls, like plants and fungus, but others do not. Like animal cells do not have cell walls. They need to be a little bit more flexible. We'll talk a lot more about the organelles later, but let's take a brief stop and look at size comparisons here. So this thing right here is part of a eukaryotic cell. And then we have our bacteria right here, our prokaryote. And then we have a very large virus right there. So just that gives you a brief size comparison between those. You've got your atom right here, proteins and lipids, tiny little poliovirus, some larger viruses that are nearly the same size as bacteria, a tiny little eukaryotic cell, the red blood cell, which is almost the same size as some of our bacteria. And then you have your eukaryotic cells right here. So just some brief size comparisons there. This is a white blood cell, a red blood cell, and this little guy in blue is a bacterial cell. So let's talk now about the organelles that make up our eukaryotic cells and get into some of their functions. So be able to describe the role of the organelles and identify the main differences between plant cells and animal cells. So first, let's start with the nucleus. This is the main dividing difference between our prokaryotes and eukaryotes. So the nucleus actually has 2 layers of membrane. We call it the nuclear envelope. So it's just an outer membrane and an inner membrane that then protects the DNA inside. And they have tiny holes on the surface of that membrane called nuclear pores. This allows chemical messengers to come in and out. It allows RNA to leave the nucleus, which is going to be the kind of middle step between converting our DNA into protein. And then inside, there's a dense area called the nucleolus. And this is where our ribosomes are made. Next to the nucleus, so we can see we've got the nucleus right here. Next to it is our endoplasmic reticulum, and it is always found next to the nucleus because it's actually part of that outer membrane of the nucleus. That outer membrane keeps kind of twisting and growing and pushing itself out into the cell. And this structure is made up of 2 different components of membrane, we can say. A rough endoplasmic reticulum and a smooth endoplasmic reticulum. You can see the rough right here, and you can see the smooth right here. Let's get into the difference of those 2. So the rough ER has ribosomes covering its surface, all these little dots all around, and that's what makes the surface of it look rough. Now this organelle, the main function is to make proteins, because it has all those ribosomes on it that make proteins, and send those proteins to the cell membrane. The smooth ER has no ribosomes. That's where it gets the term smooth, because it doesn't have that rough appearance, that bumpy appearance. And it makes lipids or fats, mainly things like hormones and stuff like that for the cell. And it also helps detoxify the cell. Any toxins that comes in get passed through there and kind of cleaned up and help the cell that way. Kind of similar in appearance to the smooth ER is the Golgi apparatus, made up of a bunch of Golgi bodies. And they look like these tiny, flattened membrane pancakes. Now, the Golgi kind of acts like the post office for the cell. This is where chemicals come in, they get processed, and then the Golgi sends them where they need to go in the cell. So they receive proteins and lipids from the endoplasmic reticulum and from other areas of the cell, and they package them up in these little membrane bubbles called vesicles. So this is a vesicle right here. All of those are vesicles. And the Golgi has a face. So it has a cis face and a trans face, in that one side is where materials come in, they get processed as they move through, and then they send them out on the other side. The next is a tiny little vesicle that is given a name. It's called the lysosome. And these little vesicles are filled with enzymes that help digest, sometimes with acid or hydrogen peroxide, and things that basically can break down other cells like bacteria or other chemicals that we then need smaller in order to process for food. So you can kind of think of the lysosome as the cell's stomach. It's going to digest and break down materials. These can also be worn out parts of the cell itself. So the cell will recycle anything that breaks down and reuse the components to help build itself up in other areas. The cell does not like to waste anything. If it doesn't have to go out and remake something from scratch, it won't do that. It'll just recycle what it already has. And they will often fuse with other vesicles that are containing like bacteria that the cell swallowed or debris, and then it'll dump all of its digestive contents into this vesicle and digest them. So vacuoles are kind of like the lysosomes of the plant cell. And we used to believe that they're only found in the plant cell, but that's actually kind of changing. We just see them most commonly in the plant cell. So are most commonly found in plant cells. I always keep forgetting to change that on this one. I need to just do that eventually. So not only, just most commonly. And they often store water or kind of parts of the cytoplasm. And in plants, we often call that the sap of the plant. So this is where it stores a bunch of that sap. And in plants, it also helps maintain a proper water pressure inside the cell. You can store dangerous toxins in there as a plant, and this is where if an herbivore comes along and bites into your leaf and breaks a bunch of your cells, they can break that vacuole and all the toxins can come out and end up poisoning the herbivore that's eating you and making them not want to eat you again in the future. And then we get to the cytoskeleton. Cyto means cell, so this is literally just the cell skeleton. These are proteins, but unlike our skeletons in our bodies, we can actually build up the skeleton and tear it down to make new pathways and new beams for support. We also use these as superhighways to move materials across the cell. We use them to help stabilize and pull apart our DNA when we copy our DNA. So calling it the cytoskeleton is a little bit misleading. Yes, it's used for structure, but it's also used for so many other things. Then we have the mitochondria. This is a small cell-shaped organelle, and we will get into later why it is cell-shaped. We'll talk about that in class. And its main job is to produce energy for the cell. And it does that through a process called cellular respiration, which we will talk about in a lot of detail in another unit. It has a double membrane, so it has one membrane on the outside and then another membrane that has all these folds on the inside. And it's all those folds on the inside where the reactions take place, these energy producing reactions. And mitochondria are found in all eukaryotic cells. They're found in plants, they're found in animals, they're found in fungi, they're found in protists, they're found in all eukaryotic cells. But only eukaryotic cells because they are made of membrane. And that membrane, remember, makes it a membrane-bound organelle. And our prokaryotes do not have membrane-bound organelles. And why are these found in all cells? Well, because all eukaryotic cells need energy. Prokaryotes have a different way of getting their energy. They still get energy, just not from the mitochondria. But all eukaryotes get it from the mitochondria. Then we have chloroplasts, and these are specialized organelles in plants that help do photosynthesis. So this takes sunlight, captures that sunlight, and converts that solar energy, that photon, into electrical energy and then into chemical energy. And that chemical energy is stored in the form of a sugar, and that sugar then becomes the plant's food, or it can be built up into the plant's body. So the chloroplast is basically a mini candy factory because it's constantly making sugar. It actually has 3 membranes, not 2. It has 2 outer membranes and then an inner membrane that forms these really interesting stacks that are connected to each other. And they look like Scrooge McDuck just went in there and made a pile of all his coins all over the place. And it's in these coin-like membrane structures where all of the reactions are going, or where the important reactions are going to occur. Some of it also occurs in the cytoplasm material outside. So that is it for the cell's structure. We're going to have one more video kind of related to this same topic that goes over the immune system and the germ theory of disease. So go watch that next.